Okay, this is a bit of a review for your final exam. There will be 3 online lectures, one for each unit, so you can pick and choose what you want to listen to. I highly encourage you to take your time, pause the video when questions come up, really test yourself. The only way to really know what you know and what you don't know is to practice testing yourself. Okay. So just by listening to this video, taking notes is not the most effective way. It's really good for you to try to practice getting that material out of your brain and seeing what you still need to study. So with that, let's start with Unit 1. So Unit 1 focused on parts of the cell, prokaryotic versus eukaryotic, animal versus plants. So I have posted, along with this video on Canvas, a chart with you can use as a quiz. So there's random blocks left blank under the cell part description function type of cell it's found in, and this is a great way for you to quiz yourself to see if you're remembering all these different parts of the cell. You want to know your functions. You want to know what types of cells they're found in. So what's similar and different between animal and plant cells and prokaryotic and eukaryotic cells. This is a great short overview video of all the parts of the cell. I'm not going to go through those, but here is ways that you can test yourself. So I have for you next a couple clicker questions. Of course, we're not using clickers, but you can practice. So what I suggest you do is you look at the next 3 slides. Hopefully you have the printout in front of you or on your screen, and you pause between each slide and see if you can answer the question. So the first question asks, eukaryotes, such as animals and plants, differ from prokaryotes in that eukaryotes what? And the correct answer is D. Eukaryotes contain numerous membrane organelles, like the rough ER and the Golgi and lysosomes. The reason A is not true is eukaryotes do have a true nucleus. That's what differentiates them from prokaryotes. All cells use DNA as their genetic material. This is not true for any cells. Cells don't use their cell membrane, but remember that eukaryotes use the rough ER for making proteins sometimes. Prokaryotes make proteins on free-floating ribosomes. And both eukaryotes and prokaryotes can do cellular respiration. Eukaryotes do it in the mitochondria. Prokaryotes do it on actually their cell membrane. Next question. A cell accumulates lipids that would normally be broken down into other cells. That does not make sense. That should be normally broken down into other... What? Let's just say, sorry, a cell accumulates lipids that would normally be broken down. What organelle is not functioning correctly? The answer is the lysosome. So remember that the lysosome is what breaks things down. The smooth ER is where lipids are produced. Sorry about that confusion. Third question. You put a cell in pure water and the cell becomes rigid or shows turgor. This is because the cell is a blank and the blank is filled with water. So when you did this, hopefully you picked This is a plant cell, and the central vacuole is filled with water. Okay. So remember that plant cells like to be extra full. Okay. Pure water would lyse an animal cell. And that, yes, plant cells have a cell wall, but the cell wall is not what is filled with water. The cell wall definitely helps the plant stand up and be in that rigid or turgor state, but it's the central vacuole that's filled with the water. Okay, so that's a few questions to get your mind thinking about parts of the cell. Another important concept we talked about in Unit 1 was the theory of endosymbiosis, right? And remember that this is how we believe our mitochondria and chloroplasts came to be. That they were once free-living prokaryotes, so bacteria, and that they were engulfed into a larger cell, and instead of being destroyed by, say, the lysosome, they coexisted. So they have inside symbiosis working together. And I want you to think about what is the evidence that we can see today. I want you to think about 3 things. So go ahead and pause the video. And hopefully you came up with some of the evidence is that both the mitochondria and the chloroplasts have a double membrane. Right? Our other organelles don't. Lysosomes, Golgi, things like that. So we think that that's because as our eukaryotic cell engulfed it, it created this second membrane. Mitochondria and chloroplasts both have circular DNA, which is very similar to the DNA of prokaryotic cells. Remember that eukaryotes have linear DNA. We have long linear chromosomes. And the third evidence is that the ribosomes, so in fact mitochondria and chloroplasts not only have their own DNA, but they synthesize their own proteins. And the ribosomes are similar to prokaryotic structure, or I should say prokaryotic ribosome structure. So it does the same function in protein synthesis as the eukaryotic, it's just that the small and large subunits are slightly different. Okay, then we talked about different types of macromolecules, and we're going to focus on lipids here so that we can connect that to membranes. So 2 really important terms to remember is saturated and unsaturated when we talk about fatty acids. So the examples here are called triglycerides. This is not what makes up your membrane. This is from food. And this is just used as an example to illustrate the difference. So saturated fatty acids means it's all carbon-carbon single bonds. And yes, lots of hydrogens around it. We're not going to draw them all. But what happens with saturated is that they can pack tightly. And this is going to be important when we talk about membrane fluidity. So this, remember, is just an example so you can visualize that saturated fatty acids pack tightly, these long carbon chains, and it results in butter. Okay, at room temperature, saturated fatty acids are what we call a semi-solid. And you can imagine you don't want your membranes to be this solid, right? Because we're going to talk about things have to be able to go through our membranes. But you also don't want them totally liquid. So unsaturated is unsaturated due to carbon-carbon double bonds, which might not mean a lot to you, except that you remember that these add bends or kinks in the fatty acid tails or the carbon chains. So that makes it more, gives it more room and that makes it more liquid. So for a membrane, we want it somewhere in between. So let's look at phospholipids. Phospholipids only have two fatty acid tails. So I just want to go back and contrast. Triglycerides have 1, 2, 3. Okay, so this is found in your foods that you eat, but phospholipids only have 2. Remember, and we draw them with a little head and the 2 tails. So phospholipids make up membranes. They are the main component of membranes, and that means the membranes around your cells. That means the membranes for all of your membrane-bound organelles, like your Golgi, your lysosome, your vesicles, your ER. All of those are a bilayer, which means 2 layers of phospholipids. And I want you to remember that in here, this part we say is hydrophobic. It doesn't like water because this is the really lipid part. And if you think of the old saying, oil and water doesn't mix, right? That's because oil or lipids are hydrophobic. They don't like water. And the heads here are hydrophilic, which literally means water loving and water fearing. So when you put the 2 hydrophobic parts together, we make this nice barrier. And the heads can interact with water. So we have water on the outside and the inside of your cell. And that's important because you need your cells to have the fluid, the cytoplasm inside, so things can move around and reactions can happen. So what does that have to do with saturated and unsaturated phospholipids? So if we look here again, and here's a saturated one, right? All single carbon bonds. It makes this nice long tail. Here's an unsaturated with the carbon-carbon double bond that puts a bend or a kink. So if we look at membranes, where we have the unsaturation, it becomes more fluid. And where we have lots of saturation, it becomes more viscous, or we could just say non-fluid. It's like thicker. Right? So we want to have this nice mix so that our membranes can have that right fluidity to allow things to cross. So membrane fluidity, let's talk about that next. Let me put this up here first. Membrane fluidity is influenced by 3 things: temperature, saturated versus unsaturated fatty acid or phospholipids, sorry, and the amount of cholesterol. So we've talked about the saturated and unsaturated fatty acids. Saturated is going to decrease fluidity. Unsaturated is going to increase fluidity. What about temperature? So if we increase the temperature, we increase the fluidity. Because as things warm up, they have more energy and they can move around, and so you get more space happening. And just the opposite, as you decrease the temperature, you decrease the fluidity, and things become more compact and more tight and more rigid. So what about with cholesterol? Okay. So without cholesterol in the cold, your membrane is not going to be as fluid. Right? So what you want to do in the cold is if you add cholesterol, you increase fluidity, right? So you put the cholesterol in here and it makes more space. So cholesterol takes up space. What do I want to say? No, I don't want to say cholesterol takes up space. Cholesterol makes the phospholipids move apart. That's what I want. The opposite, if you have a membrane in a very warm environment, it's going to become too fluid. The molecules are going to have too much space. So now if you add cholesterol, you decrease fluidity because cholesterol is taking up space. Right, so you interject cholesterol in here, and now your membrane isn't too fluid. Okay. So cholesterol's effect is dependent on temperature. Temperature, cold decreases fluidity, hot increases fluidity. Cells can't control their temperature, but they can control their amount of cholesterol, and they can control the amount of saturated and unsaturated fatty acids on those phospholipids. So let's do a couple questions and see if you're getting these concepts. So again, I encourage you to pause the video. This first question asks, which of the following words best describes, so many typos, best describes the fatty acid shown below? Hopefully you chose saturated. These aren't kinks. Remember, these are just showing how the carbons are hooked together. So this is saturated. And hopefully you chose the word hydrophobic. So we're going to talk about polar, and yes, there is a little bit of polarity here, but the majority of this molecule is hydrophobic. Next question. To increase membrane fluidity in the cold, a cell could, and then I want you to think about why. So pause the video, and then when you're ready, let's do the answer. So the correct answer is A and C. So if we were in the cold, if we increase the amount of cholesterol, right, That's going to push space and make it more fluid. The other thing we'd have to do is increase the amount of unsaturated fatty acids. So we want those fatty acids with kinks in the tail that are going to take up more room to increase fluidity. These are things that a cell can control. They can't control temperature, right? B is not correct because if we increase the amount of saturated fatty acids, we're going to make that membrane more compact, which it already is more compact in the cold temperature. So we don't want that. Okay, your last question for this section. Which of the following statements describes the structure-function relationship for a phospholipid? So, lots of words. Pause. And so the correct answer for this is B. The hydrophobic tails and the polar head groups allow self-assembly into a bilayer structure. That's the basic structure of cell membranes. Right? So the phospholipids, the tails go together to create this nice barrier. The heads interact with the water environment. Okay, so let's say why these other ones aren't correct. Okay, so we don't join the tails together by dehydration synthesis, and we don't want a thick, rigid membrane. No, that's not good. Hydrophobic head group can associate with the polar tails of other phospholipids. So this is kind of what this thing is talking about, and that's not how a bilayer is formed. The polar head groups associate with one another in a tails out structure. Well, that wouldn't work because there's water. Remember that you are mostly water, and those tails do not want to be around water. They want to be away. So that's why they make this nice layer. So that's not correct. That one's not correct. B is correct, so E is not correct. All right. Let's talk more about those phospholipid bilayers. So remember that here's again our phospholipid, and because we talk about its hydrophobic and hydrophilic, we also use the term polar, which means it can associate with water, and non-polar means it doesn't have any partial charge. And I want to remind you of the word amphipathic. This molecule is amphipathic because it has both properties, both a hydrophilic part and a hydrophobic part. All right, let's talk about these very important words. Semi-permeable versus selectively permeable. Semi-permeable. Semi means some things can cross. Let's not say things, let's be a little more specific. Let's say some molecules can cross the membrane. That means they can go right across this membrane. I guess I shouldn't draw it through a phospholipid. They're going to go right through the space between. That's semi-permeable. What can cross the membrane like this? Gases. Oxygen and CO2 are the big ones we talk about. Water. And hydrophobic molecules. And sometimes this is counterintuitive because we have this hydrophilic head, right? And here. But because the majority of this membrane is hydrophobic, other hydrophobic molecules, remember those are lipids, can pass through, and the main one we usually talk about is steroids, which are some of our hormones, like estrogen and testosterone. Remember that cholesterol shown here is also a steroid, but it actually hangs out in the membrane. Okay. So, semi means some things can cross. Anything that's crossing through the semi-permeable, this is called simple diffusion. And we'll talk about these terms again, but I want you to know semi-permeable and simple diffusion go together. Okay, I'm going to erase the information on this slide and we're going to talk about selectively permeable. Okay. Selectively permeable means the cell, or the membrane, I should say, can control what crosses. It selects. This requires proteins. So while semi-permeable allowed things just to cross right between those phospholipid molecules, selectively permeable, something that controls, you have to have a mechanism to control. And those are proteins, and so this means that we're going to be talking about facilitated diffusion and active transport. Okay. And this is how most molecules cross the membrane. They need some kind of protein. And what's the difference between this channel protein and carrier protein? Remember that, I'm just trying to get my change color. A channel protein is like a tunnel. It's open. Sometimes it has a gate on it, but it's just basically a passageway through. Still very specific. And if you could look at the molecular structure in here and the amino acids, because now you know that proteins are made of amino acids, you would understand that the amino acids interact with molecules and will let some through and not others. A carrier protein is something that changes shape as molecules cross the membrane. So I want you to think of it just like if you were carrying something, right? You've got to pick it up, you've got to bend and put it down, right? Pick it up, put it down, or pick it up and move. Pick it up from the floor, put it on the table. Even moving from one place of the table to another, you have to change your shape. You have some kind of motion. So that's really the big difference between channel and carrier proteins. Both can be part of facilitated diffusion. For active transport, we are using something called a pump, and that's a type of carrier protein. Okay, so semi-permeable, selectively permeable. All right, let's talk about what can and cannot cross the membrane, and then we'll get back to our transport. I want you to remember what it means to be polar or nonpolar. Polar means there are slight charges or partial charges on the molecule. Okay, so here's water. This is our classic polar molecule. Oxygen has a slight negative charge. excuse me, hydrogens have a slight positive charge. And that's because oxygen likes to hold on to the electrons a little bit more than it allows hydrogen to. This is chemistry that we didn't really get into, but I want you to understand why it has a slight charge. It's not an ion. It's not sodium plus. That's an ion. That's not what we're talking about here. So because water is a polar molecule, other molecules that are polar like to dissolve in water. What you're looking for is OH groups and, shown here, NH groups. These are the polar parts of biological molecules. So we'll see these on proteins and sugars and even DNA and RNA, our nucleic acids. This molecule over here is nonpolar, which means there's no charge, not even a partial charge, and it does not dissolve in water. So the classics that we're looking at are lipids. Okay. And I forgot to put over here, the nitrogen would have the partial negative and the hydrogens again would have the partial positive. Okay. So if I ask you if a molecule is polar or nonpolar, you're going to look for a bunch of OH groups or NH groups and you'll know it's polar. If it has only one or two OH groups and a lot of carbon-hydrogen bonds or carbon-carbon bonds, remember like we just drew on our fatty acid tails, that's nonpolar. And I just want to show you what it means. If something's dissolved in water, it means that the water is surrounding it, right? So this is how we keep our cells fluid. This is how we allow all the molecules to move around. So this is just showing you some sugar molecules. They're the big hexagons surrounded by water. And again, when you're looking at this sugar, you would know it's polar because look, it has 1, 2, 3, 4, 5 oxygens, and 5 OH groups, and another oxygen, and it's not a long chain of just carbons and hydrogens. That's really what you're looking for. So this is polar. So let's talk about our transport again, right? So transport across the membrane, we have both passive and active. Passive is saying that a molecule is moving from high concentration to low concentration. Right? And these are all showing high to low. Watch, see the flow of the arrow. We can also say a molecule moves down or with the concentration gradient. Those are all saying high to low. Okay. That's all passive transport. If we call it just diffusion, it's implied it's simple diffusion, which means it's moving directly through the membrane. From high to low, and we said this was like gases and water and hydrophobic molecules. If it's not one of those, and it needs some help, it's facilitated diffusion. So this is diffusion that requires a protein. But they're both moving from high to low concentration, and there's no energy required. Let's contrast that to active transport. Okay. Active transport is going against or up the concentration gradient. Which means it's going from low to high. So just look at the orange here. There's 1, 2, there's 1, 2, 3, 4, 5, right? So it's going from low to high. And the key here is this requires energy. It's harder to do, and it requires a protein. So active transport always requires a protein. We call that protein a pump. So if you see that word, or if you see it's talking about requiring ATP, you know it's active transport. Okay. The other one that's super important, and I hope you remember this when you get into GB2 and into cell biology, is what's called secondary active transport. Okay. And this means that one solute or molecule is moved against its concentration gradient by using the energy generated from another one. Okay. So we have to have primary, which is what we just talked about, to drive secondary. So let's break this down. Okay, so if we look at this first part on the left, we have the sodium-potassium pump. And the cell is pumping using ATP, so this is primary transport. Primary active, sorry. Oh, I can erase this. Okay, it's using ATP and it's making sodium a high concentration up here. Now, the secondary active transport is over here. And that's where glucose is being moved from low to high concentration, which we know requires energy. But instead of using ATP, it uses the energy by a gradient of another solute. So it's using the energy of Na+. So Na+ is now moving high to low. And it's bringing with it glucose going low to high. So glucose is being moved by secondary active transport. There's no ATP in secondary. Okay. Primary uses the ATP, creates a concentration gradient, and brings something through it. And hopefully this is clicking with you. Oh, we talked about in cellular respiration and in photosynthesis an H+ gradient that can drive the formation of ATP, right? So electrons gave energy to pump the H+ across, then that H+ was used as an energy source to come down its concentration gradient and do something. So this is transport because we're transporting something. The one I just talked about in mitochondria and chloroplasts is chemiosmosis. So take a look at this. Again, sodium is being moved by primary active transport, which requires ATP. Glucose is being moved by secondary active transport, which is using the sodium concentration to move that solute. Okay, we have 4 clicker questions coming up. The first one is asking you, a large polar molecule can move through the membrane down its concentration gradient by, so pause the video, figure this one out. And the answer is facilitated diffusion. 1, it's a large polar molecule. We know that can't go through simple diffusion. Only very small polar molecules like water can. It's going down its concentration gradient, so we know it's some kind of diffusion. A molecule that moves against a concentration gradient without the use of ATP moves by, and hopefully you came up with secondary active transport. So this was how we were moving glucose. Glucose was being moved against its concentration gradient, so low to high, but without ATP. It was using the energy of that sodium concentration gradient. That's secondary active transport. This molecule will cross the membrane down its concentration gradient by, hopefully you recognize this as a steroid, which is a lipid, and that means it goes through simple diffusion. So I know this gets a little confusing because there are 2 OHs, but there are so many carbons. So much more than you saw in that glucose molecule. If you flip back, right? This has 1 ring of carbons and hydrogens. This has 4 rings. So even though there's a little bit, there's so much carbon and hydrogen. This is a hydrophobic molecule. It can go right through the membrane by simple diffusion. Okay, your final question is, which of the molecules below are polar? There's more than one correct answer. So pause the video and figure this out. And hopefully you recognized that A, B, and D are all polar. So look at all these OHs. Okay. And oh, we even have a charge here, and we have NH, and we have OH, and we have another OH. This is a nucleic acid. This is DNA. Well, a DNA nucleotide. This has got NH and OH. This is an amino acid. That is also polar. But this one, even though there's some oxygens, look at all these carbons and hydrogens. Okay, this is a lipid. All right, a couple more concepts and that will be the end of Unit 1. So osmosis. Remember that osmosis is the movement of water from high water to low water concentration. So it's like diffusion of water, just gets its special name. Why do you think water has 2 ways to cross the membrane? So water can cross by simple diffusion and facilitated diffusion. So water can, this is showing you simple diffusion. It's going right across that membrane. Okay. That's simple diffusion. Water can cross that way, but it's pretty slow, not super efficient. Okay. So facilitated diffusion, Many cells have something called an aquaporin, which basically means a water tunnel. And that's this water selective pore. And look how much water can go through. So water can move much faster through facilitated diffusion because it's got this nice opening, this nice tunnel for it to go through. Okay. But it can also move by simple diffusion. So water is essential to life. We do not want to block water out. We want it to be able to move across membranes for wherever we need it. The other concept that goes along with water are the terms hyper, hypo, and isotonic. And we're talking about solutions. So we're talking about this liquid that a cell is in. Okay. So that could be your blood. That could be your lymph system. You have lots of solutions all over your body. The tonic part, the tonic refers to the solute, which you can just remember as the stuff in the solution. So this could be salts, this could be sugars, this could be proteins, this could be lots of different molecules. So what's shown here is in brown is our cell. And I'm going to ask you what type of solution the cell is in. And so we're looking outside the cell. Hypo means low or less. Less stuff. So I want you to see that there's only one green dot here, multiple green dots inside the cell. So this is a hypotonic solution. There's not a lot of stuff in it, which means if there's less stuff, there's more water. So a hypotonic solution has a lot more water than inside the cell. Just the opposite with a hypertonic. This means lots of tonic stuff, which means that there's less room for water. So a hypertonic solution has a lot of stuff outside, and there's not as much water, so there's more water inside the cell. And isotonic means same or equal, and that means that there's the same amount of stuff on both sides and there's the same amount of water on both sides. One thing I want you to remember is that water is always moving, right? Even in an isotonic solution, it just means that the same amount of water that comes in the cell goes out the cell. In a hypotonic solution, because the water concentration is much higher, water is going to move into the cell from lots or high water to low water. And in a hypertonic solution, it's just the opposite, right? And you don't have to memorize this. You just have to know the key words and you can figure it out. So let's do a couple clicker questions and see if you've got these concepts down. And then you can go on to the second video if you like. So what type of solution is this cell in? So go ahead. I would pause this the video, answer the next couple questions, then come back. So what we're doing is the cell, this is my cell, this little squiggly thing, and it says there's 20% of salt outside in the solution, and that is greater than the 10% salt in the cell. So this is a hypertonic solution. There is more here than inside the cell. Same scenario, the next question is asking which way will the water move initially? So if the solution has 20% salt, it's going to have 80% water. And for the cell, it has 10% salt, so it's going to have 90% water. Water is going to move from high water to less water, so water is going to move out of the cell. Okay. Eventually water will reach equilibrium and it will equally move back and forth, but initially it's going to go from high water to low water concentration. Okay, your last thought question for this unit says reverse osmosis is a method of making fresh water. Based on the image below and what you know about osmosis, which way will the water move in reverse osmosis? And so the answer is reverse osmosis means the water is going to move to the right. And this does take energy. You can see we have applied pressure. So even though this is higher water concentration in fresh water, normally the water would move this way, right, to the left. But in reverse osmosis, what we do is we basically push out the pure water and leave all that salt and contaminants behind. So it's a really cool kind of like, you can think of it as like an active transport of water. A way for places without a lot of fresh water, with a lot of salt water, to create pure, fresh drinking water. Okay, so that is the end of Unit 1. The next video will go through energy, enzymes, photosynthesis, and cellular respiration. Oops, and I don't know how to turn this off.